I think we're going to cover COVID-19 vaccines and their boosters this morning. We'll let people trickle in slowly while I'm giving some introductory thoughts here. Javel knows this. This is the third version of this talk that I'll be giving, and every time I give it, I always think to myself, well, surely that's enough and we're done with it. And then new things happen with vaccines and new things happen with boosters and all that kind of stuff. And so some of the slides, I last gave this talk September of last year, right when we were talking about first round of boosters. There's a lot of different information in it for sure. But if you saw that talk last September, some of the slides, introductory slides, are going to be familiar. And so most of that is going to be in giving background. As always, it's easier for me if you have a question, if you just unmute yourself and shout it out. But Kayla is going to help watch the chat, and we'll jump in if necessary, if chat is the easiest way for you to ask the questions. So that we're just going to go ahead and launch here. So we're going to cover a lot of ground here. We're going to talk generally about COVID-19 vaccines. We're going to go over some information about the mechanism of how they work, just as a reminder of the biology of how they work. We're going to cover some of the important information about how we think about efficacy vaccines and how we deal with that. We're going to spend a little more time talking about how vaccines are approved. And the reasons that we're going to do that is because what we're going to do as an exercise in the second half of our session is we're going to play the part of the approval bodies. We're going to play the part of the CDC and the Advisory Committee on Immunization Practices and pretend that we are making the decisions about whether to approve bivalent vaccine boosters. And I'll share with you at that time all the data that they had. It will literally be the same exact data that they have. And then we're going to ask questions and think about it and have a chance to vote as if we were on the ACIP. And then I'll share with you a little bit about how the ACIP voted and what their conversation was like. And I'm curious to see whether our conversation is going to mimic theirs. So we'll talk a lot about the factors that are weighed in considering vaccine approval and the data and the side effects and those sort of things. And then time permitting at the end, would like to spend some time talking about how do we address vaccine hesitancy. Again, we've talked about it before in Academic Half Day, but many of you are new this year. And I think that some of the issues around vaccine hesitancy, while they may be the same, some of them are different at this point in time since last year. So let's go ahead into it. So the overall objectives, so what I hope you get out of it, besides going through it this way, is I want to hope that you get out of it a good review of what the mechanisms of action are for the current vaccines. And then be able to describe to others what the regulatory process and approval process is here in the United States. And then do a little critical appraisal about the evolving data related to bivalent vaccines so that you can advise your patients and provide the best information for your patients. And then again, time permitting, have a chance to practice some scripts or develop some scripts so that you can help perhaps address some of the vaccine hesitancy among your patients. Okay. All right, so COVID vaccines. COVID vaccines, most of what we're going to end up talking about in much of this talk is the mRNA vaccines. I will touch on some of the other vaccines just because it's useful to know what's available around the world. And also in the rare instance, but there are still a fair number of people within the United States who've gotten some other vaccines. And so let's talk about those. The main mRNA vaccines obviously are the ones from Pfizer-BioNTech. The trade names are Comirnaty. If you're wrapping your head around why Comirnaty, it's because the RNA is in the middle of it. And so that's their way of saying that it's an RNA vaccine, but it is a little hard to get off the tip of the tongue. And then Moderna. Moderna's trade name is Spikevax, which sounds like something that you bring to camp and play against spiking the ball down and playing whatever that is. There is a third vaccine that we're just going to briefly mention, and I'll talk about why it was a failure, which is CureVac. And CureVac was an attempt to also make an mRNA vaccine for much cheaper than the other two. Okay, so what are mRNA vaccines? So remember that the coronavirus here is a positive expressing mRNA virus. And so what the researchers have done is taken the RNA sequencing from the original ancestral, even though it's only a few years old, version of the virus, the wild type. And this comes from the Wuhan province. And they identified the segment that encodes the spike protein. And what they've done is they've taken that, they've made some alterations to the mRNA fragment and placed it into lipid nanoparticles. The lipid nanoparticles are then what gets injected into the body and into your IM area. And those vaccine particles then, with the lipid surrounding, then fuse with the cells, and the RNA segment does what RNA does, and it gets translated. And the spike protein that is produced out of that is a trimer. It's a three-part protein that collects together and ultimately form the spike that is the protruding spike of the coronavirus. And so then this is displayed, and your body, doing what it does, will then take as those fragments. The antigen presenting cells then will do its thing and present it to B cells and helper T cells, and that's how you get the secreted antibodies. You all are much closer to immunology than I am, but hopefully this sounds very familiar to you. Then when you are exposed to another infection, then what happens is that the B cells do their thing, produce the antibodies, and will then your body's immune defense system will do its thing and destroy, hopefully, the virus. Okay, so that's the mRNA vaccination. Pfizer and Moderna have some slight differences. They essentially encode the exact same segment of the mRNA fragment, but there are some small differences, and they turn out to be perhaps important to how they perform. The main differences are that Moderna, the dose for the primary series is 100 micrograms versus Pfizer's version, which is 30 micrograms. And so if you've ever heard anecdotally that people are having more side effects for Mrna than they are for Pfizer, in fact, it turns out to be true, that the data support that. And that's probably largely because of the difference in the doses between the two. Now, Moderna's booster doses, and we'll talk about both the bivalent and the earlier version of their booster doses, they took that and they halved it. So it became 100 micrograms, went down to 50 micrograms for the booster, still more than the 30 micrograms. The Pfizer boosters are 30 micrograms. And then the other big difference is, in the primary series, when we went through this, is that Pfizer was licensed as a 3-week interval and Moderna was licensed as a 4-week interval. And it turned out, and the immunologists knew this all along, it turns out that actually your body does better when exposed to a second exposure to things if it has a little time to mature and think about it and develop a chance to develop those reactions. And so in many ways, the early data around Mrna ended up looking a little more favorable, and I'll show you some data that confirm that this is true in terms of some of the early early results from Mrna versus Pfizer. I mentioned that there was another company that was trying to make an mRNA vaccination for much cheaper. They had some theories on how they were going to do their vaccine, and they went with a much smaller dose, 12 micrograms. They also did something which is the Pfizer and Mrna, instead of using uracil as one of their bases for their mRNA segment, they used an altered form of uracil, and the CureVac people used the natural uracil. Well, it turns out that in your body doesn't like mRNA circulating in your blood, right? I mean, that looks like an invader. And so your body actually has mechanisms to break down mRNA fragments that are loose. The pseudouracil that is in the Pfizer and the Mrna vaccines actually slows that process down so that it stays in the body a little bit longer. The CureVac people used natural uracil, and the idea was that actually they thought maybe that would provide some inflammation that your body could use to stimulate the immune response. CureVac turned out to be a wild failure, and the company is no longer producing the vaccine, and its data did not work at all, and it was a huge disappointment to a lot of people. All right, so that's the mRNA vaccines. Hopefully that all sounds relatively familiar in thinking about mRNA. As I said, mRNA is not very stable, and you remember that Pfizer and Moderna's versions of the vaccines need to be actually kept at very cold temperature because they're not stable. mRNA as a single strand tends to break down much quicker than DNA. DNA is much more stable, and so as you know, a lot of researchers, particularly those with Oxford and AstraZeneca and Johnson & Johnson, decided to go a different route, which is to use DNA, and DNA being more stable, they thought this might actually be a way to do this at room temperature and to create a more stable version. And so what they did is they took the positive sense mRNA strand, they created a double-stranded DNA version of that, put it into adenovirus. They chose adenovirus vectors that were immunogenic, but did not have infection risk because of the way they changed it, to create problems in people. And so that DNA segment then fuses into a vaccinated cell, does its thing in the nucleus. It then creates the mRNA segment that is used, and then the rest of it looks the same as the RNA vaccines. So the adenovirus that they chose, so the AstraZeneca people chose a chimpanzee version of adenovirus, so something that normally doesn't cause respiratory infections in chimpanzees. And the Johnson & Johnson people basically took a version of an adenovirus and did some changes to make it incompetent to replicate on its own. There's a Russian version of this. It was called Sputnik 5. I'm not sure what Sputnik means in Russian, but it was their version of this. What they actually ended up doing, it's probably by accident, they did not create a completely replication incompetent version of the adenovirus, and it ended up infecting some people, immunocompromised people, with adenovirus, and it created some problems, as you can imagine. The other thing that they did, and if you have wondered why AstraZeneca is not approved here in the United States and it is in Europe, they chose, AstraZeneca and Sputnik chose a different version of the spike protein. And what this means is the coronavirus, when it has its spikes that it's working with looking for ACE2 receptors, that spike protein looks a certain way, like you see on the left. After it fuses with the ACE binding sites, then it changes its conformation and the spike elongates to the right side there. And so not entirely on purpose, but because it was what was available, the researchers for AstraZeneca chose the post-fusion spike conformation for what they were encoding for. What the researchers in the United States did, there was a group of researchers that actually figured out, and they did this because they were doing research on other viruses, other respiratory viruses in children. What they figured out is that they made some small changes to the conformation that kept it in this shape, the pre-fusion spike shape. So if you did sort of a thought exercise and said to yourself, what would be better to help prevent circulating virus? Is it the virus after it's changed its shape and already fused in the cell, or that before it has? And so there's reasons to think that the shape of the spike protein that they're coding against makes a lot of difference. And in fact, the results for the AstraZeneca virus vaccine are not as good as the Johnson & Johnson version of this. Okay. And if you're curious, Moderna and Pfizer also use the pre-fusion version of the spike protein, and so probably makes a difference. Okay, I know I'm moving quickly, but let's talk about the last group that's approved in the United States, and this is the most recent group. So Novavax, I'm sure many of you have heard about Novavax, but then it kind of disappeared off the radar for a long period of time. There's a lot of excitement about this. Novavax is a protein-based vaccine. it's done in much more of what we might think of a traditional way, in that many of our vaccines historically have basically been proteins or subunits of proteins rather than the DNA or RNA that encode those. Um, theirs was finally approved and finally licensed, and as of at the end of August, there were a fair number of people within the United States that had received this. The idea being that there were some people who did not like the idea of hearing that a DNA or RNA vaccine was going into the body. Although not technically correct, there was also people on the internet talking about ways in which we were changing your DNA and all that sort of stuff. And so this was meant partly as a way of addressing those things. The name of it, they realized that people could not remember that Spikevax and Mrna T referred to Moderna and Pfizer, and so they actually took their name Novavax and used it cleverly in their name, or somewhat cleverly, as Nuvaxovid. Still a mouthful, but anyways. This is really kind of remarkable. The science behind this is remarkable. And so they took the RNA and encoded a DNA. And what they did is they actually used a virus vector to put this into a cell culture. And in order to avoid the risk of infecting other people, they put it into a moth virus. And this moth virus infects this culture of moth cells and use this to produce the spike protein. And the spike protein, they went ahead and collected. They added it together in these nanoparticles with basically an adjuvant. The adjuvant comes from the bark of a willow tree, I believe. And this adjuvant then is used to activate the immune system and do its thing. And so, you know, pretty crazy to think about moth cell media and all those sort of things. So it's been approved. It was approved in August. Novavax is now recommended, just like Moderna and Pfizer, as a two-part series for people aged 12 and older. As of the end of last month, a little more than 500,000 doses were given, and given across the states, but still not high uptake, right? Like you can see the numbers there, that still less than 10,000 people have received the full dose series. Okay, I'm only going to mention that early days of the virus vaccine development, there were different virus vaccines that were developed elsewhere. Sinopharm and Sinovac were done in China. They're basically doing our very, very old-fashioned way of taking virus, irradiating it, and sticking it into people. And the results in terms of the immunogenicity and how much protection was afforded by those turned out not to be that great. It turned out to not produce that great a protection, but it was cheap and it was fast, and a lot of people, particularly in Asia, got those versions, especially in the early days when we were all scrambling to try to find vaccine. What kind of vaccine is Corbavax, the patient-free one, the patent-free one? So Terrence, you'll have to remind me, because of the brand names I don't always know. This is the one that's being produced in Texas, or by Peter Hotez in Texas. Yeah, I need to look up, and I will try to get back to the group exactly what the mechanism is. What Terrence is referring to is that Peter Hotez, a scientist at I forgot, Baylor or somewhere in Houston, has produced a vaccine and essentially has put it out in the public domain for people. Yes, Baylor, put it out in the public domain and has said to essentially, particularly the global South and resource poor countries, that they can have it without needing to pay anything. And so the hope is that making it free for people, or at least the intellectual property part of it free, that we can increase vaccine uptake. But I don't actually know. I need to look up the mechanism. Okay, so emergency use authorization. Now, of course, FDA has given full authorization for Pfizer and Moderna. So as you know, there were a number of things that happened, and I'm just going to briefly touch on them just so that people can remember these. You'll remember that in the adenovirus vaccines, there was a real problem, probably not just a problem in terms of numbers, because the numbers were relatively small, but a huge problem in terms of public perception and acceptance of the DNA vaccines, the adenovirus DNA vaccines, because there's a hit-like syndrome. There's basically a very analogous condition to heparin-induced thrombocytopenia called vaccine-induced thrombotic thrombocytopenia, especially in young women. And then the other adverse event that has also been widely reported is myocarditis and pericarditis, especially in young men and boys with the mRNA vaccines. And we'll come back to this because it will be part of our discussion about safety. And then I mentioned this, that we have come to greater appreciation that longer interval between doses probably makes a difference to the immune reaction and immune response, and that higher doses may make a difference as well. The extension of the emergency use authorization, as you know, is now for children as young as 6 months. And we have, not surprisingly, discovered that immunocompromised patients don't do well in terms of production of antibodies. And so for those, the primary series now says 3 doses, not just 2, for those that are immunocompromised. Okay, so those are some things that have happened since those. Let me pause there, and we're going to get into some thoughts about efficacy and how we think about efficacy, and just check to see if there are any other questions. Cool. Yeah, very cool stuff. I'm not a lab scientist, but I think it's really amazing how fast and how quick and how beautiful the science is. All right, so I'm going to remind you of the single best graph of efficacy I've ever seen in science. This is the New England Journal of Medicine article, first reporting results from the Pfizer-BioNTech trial, looking at placebo in blue here and the orangish color here of people who received the Pfizer vaccine, and seeing that within 10 to 14 days, there's wild divergence in terms of reduction of risk of symptomatic infections. This one rightfully got everyone's attention, got everyone incredibly jazzed about that the vaccine was more wildly successful than anyone was willing to dream that it would be. And people, as you know, many people rushed to get their vaccinations. I'm going to make a parenthetical comment here that this also may be, in some ways, the source of why follow-up vaccines have been challenging and problematic for people. The vaccines largely were done, as many vaccines were, with an eye towards the goal of reducing hospitalizations and deaths. After this graph came out and after people saw that maybe we could actually prevent infections from getting this vaccine, move the goal line, to use a sports metaphor, and people had higher expectations of what the vaccine should be able to do. And we'll show some graphs of what actually happened over time. But this, I think, is probably in some ways indirectly responsible for people saying the vaccine doesn't work in the way that they thought that it was meant to, because we probably wildly oversold the promise of what the vaccine was ever meant to do. Okay. I like math. I'm not going to force you into it. I'm just going to remind you on how we calculate vaccine efficacy. The efficacy of something, an intervention, a drug, or in this case, a vaccine, is defined a few different ways, but I'm going to really focus on this bottom one here, which is to take the incidence rate in the arm and put it as a ratio over the incidence rate in the placebo arm. In its very simplest form, that's how you calculate efficacy. Okay, so in the Mrna mRNA vaccine, which had a graph that was very similarly very positive, they showed that there was 3.3 infections per 1,000 person years in the vaccinated group, and there are 56 over 1,000 in the unvaccinated group. And so if you do the math, and I've done it here, they came up with a vaccine efficacy of 94% against symptomatic disease. Okay. The problem, I think, starts from the very beginning on how do you explain what a 94% efficacy is to a patient or skeptical family member. early on in the disease, people were mistakenly describing this, saying that 94% of people would not get the disease and 5% or 6% of people would get the disease, which is not in fact what this says. I mean, this is a ratio of the expected rate versus the seen rate with the intervention. And as you might guess, it depends a lot on what the background rate is, right? So if there's a lot of infections, that there are more people who are going to get sick even among the vaccinated group, just because the infections are so much higher than if the infections are rare. And so the 94% is a relative comparison compared to what is going on in the background. So again, hard concepts. Statistics are always hard to explain to people, and for a large public, it's an even more difficult thing to try to explain. So, of course, we all know that this is not what actually happened. And to show you a little bit graphically of what happened here, and to remind you of what this looked like, this comes from a systematic review meta-analysis done. It was done earlier this year out of BMC Infectious Disease. What they did is each of these circles represents a study. Okay, and so the larger circles represent those studies with more data, the smaller ones represent those with less data. And what they've mapped on the bottom axis here, the x-axis here, is they said, okay, time matters, and what happens as time goes on, time since your full vaccination. And the vaccine effectiveness is plotted here on the left side here. And so again, early on, 95% efficacy for a lot of these studies. And what did we see? We saw that over time, particularly once you got past around 4 months, the efficacy started to drop. Okay, and so this maps with what we saw and what we saw in actual what was going on in the US and what was going on worldwide. The other colors represent the different vaccines, but overall, all of them show the same general trajectory, which is that, again, after about 4 to 5 months, the vaccine effectiveness drops below 50%. And once you get to a year or less than a year, 2/3 of a year, you're really back down to background efficacy in terms of preventing infection. Okay, so think to your patient panel, think to some of the patients that, I was looking at this yesterday in my patients that I saw yesterday, and I did see some patients who have not gotten any boosters. And so many of them who've gotten their vaccinations a year ago, they essentially have little to no protection against an infection. Again, contrast that with that beautiful picture of 95% efficacy, and you see the disconnect that people are facing and that we're facing here. Okay, what about not efficacy, but what about, again, the thing that we were really designing it towards, which is severe COVID-19. And so in this case, we actually see very durable responses, very durable protection against severe COVID-19. The different studies may have defined it in very different ways, but all of them included death. All of them included things like hospitalization or ICU stays. And so again, even now, more than 4 months, now 2/3 of a year, we have very robust protection against severe disease. I see a question, is the size of the circle the sample size? Yeah, no, so Joshua, technically it is correlated with, it's inversely correlated with standard error. And so in a meta-analysis and graphic representation like this, the larger circles are those with smaller standard errors. That the simpler way to think about it is that standard errors often travel together with the sample size of the study. And so a simpler way to think about it is larger studies versus smaller studies. And when they fitted the curve, the kind of dotted curve that you see there, the red one in this case, is a modeled curve based that takes into account those things like the sample size and how it weighs on the final number. I hope that answers your question. Okay. All right. So that's a meta-analysis of worldwide data. What about here in the United States? And so In fact, here, this is from the MMWR, the CDC's publication. And again, they basically say the same thing here. They actually did break this out into what about sort of the Delta period of time? What about the Omicron period of time? And so you'll see here 2 lines in each of these cases. So for those that received 2 doses, for those that got a booster, what is the protection like? And you'll see here in the Delta period, still 88% effectiveness, even up to, close to 160 days after their dose. And there was a bump in effectiveness, but still very robust protection. By Omicron period, again, Omicron is later than Delta, and so we see some greater time, 256 days here. The effectiveness against hospitalization had dropped a bit, and with the 3rd dose, with the first booster, you see a robust bump in that protection. Okay. Second here. Okay. So let me pause and do a thought experiment. What are some of the reasons the efficacy has changed? Why does the efficacy diminish over time against infection? Why does the efficacy against severe disease remain robust? And what are some of the possible confounding issues? And so unmute yourself, throw in the chat, whatever. Do you have any ideas or thoughts of what might explain this discrepancy between protection against infection and severe disease? What are those things that might be impacting this? Anyone have any thoughts? Evolving viral strains, yeah, big confounding issue, right? We're going to certainly talk about that. Any other thoughts? Herd immunity, yeah. So there are a lot of people who've gotten infected. There are a lot of people who've gotten vaccinated. Yeah. Changes in behavior. Ruth, thank you for pointing that out. Yes, changes in behavior. Everyone, how many have you heard your friends say, I'm done with the pandemic, right? I'm sorry to say it, we don't get to decide that entirely, but our behavior does. Yeah, Paxlovid, well, we can talk about Paxlovid. Maybe we'll save that towards the end, whether or not that's changed the curve. I'll give you the preview on that. I'm not sure that we know the answer to that one. I think that the Paxlovid issue is pretty complicated. Yeah, decreased circulating antibodies, some decreased activity in B cells over time. Yeah, Luke, I think this is what the immunologists are wondering about and thinking about. And I'm not an immunologist, but I think we'll talk a little bit about what all this means. So yeah, I think that those are all very good, very thoughtful reasons to think about. So yeah, it turns out, and we're going to come to this first point because it turns out to be very important to our studies about bivalent vaccines. It turns out that the circulating antibody levels and the amount of antibody needed to neutralize in a serum a potential infection is fairly highly correlated with how much protection you actually have against symptomatic infection. Here's the other part, and this has to do also with the changing variants, is that, in the earlier days, we felt like 5 to 7 days after exposure was about the window in which many people were actually starting to develop symptoms and becoming symptomatic. And as you remember from immunology, that it can take 7 to 10 to 14 days for B cells to increase their antibody production. And so for something that might be a longer lag time, a longer incubation period, it may be perfectly fine to have those B cells activate and produce antibodies 10 to 14 days later. But as this period has shortened, as the variants have changed and become more rapidly infectious, we see that, in fact, it's so fast that you don't actually have a chance in many cases to develop the antibodies to help prevent from getting infected in the first place. Now, it may be, in fact, that, 8 to 10 days was when we were seeing many people become a little bit sick to severe multi-system inflammatory syndrome. It often took about 8 to 10 days, and maybe that 10-day window that B cells take to produce cells, maybe that's actually enough to keep people out of the hospital. Who knows? I'm making stuff up, but I think this is what immunologists are wondering about. What are also other possible confounding issues? We're talking about this changing population behavior, so the masking behavior. Time is the biggest variable. in all of this, and the CDC has, and you'll see as we talk about this, time is a really important variable in all this. And then, as we're saying, the changes in infection potential and breakthrough potential for the variants. Okay. Ruth, yeah, more reporting and access to testing. I'm going to show you a graph. This is a recent phenomenon of this issue about home testing and what it's meant for our knowledge about what's going on. But it is, there's a real concern there. Okay. We've talked about it, we've touched on it. We can't not talk about the variant changes. The picture here is very busy, and I apologize, but what I have done here in order to create this image was take images from various sources and kind of cobbled them together into one picture here. So these are all from CDC sources, so they're all the same places. But what you see here is in January of 2021, so almost 2 years ago, not quite 2 years ago. That was, if you remember, that was when we were still in the throes of mostly original virus. You see here, as January into June, this is where we first saw alpha come up, okay? This tan color here, it got changed in the color to light blue here, but this is alpha, when alpha was predominant. What you see here, this is a proportion of the total virus. It doesn't reflect how much virus was circulating, but this is the proportion of the tests that we were doing. Then in mid 2021, this is when Delta took over, and Delta, as you know, killed a lot of people. It was very infectious, also caused severe disease, and that's when we saw a lot of Delta. So this is a slice in time, January through September of last year. What about the same period of this year? So January through September of this year, I'm missing a picture from April until June, so you have to kind of use your imagination here. This is Omicron, right? This is the Omicron era. This dark purple here, this is when Omicron BA1 subvariant was predominant. Then BA2 had a brief time in the sun here in March. And then around June, starting around June, this is when we saw the rapid rise in BA5. We'll talk a little bit about what those numbers mean and all that sort of stuff. But all of this, and this date here, mid-June here, turns out to be important for reasons I'll share with you in a moment. But all of this makes vaccine development and vaccine decisions quite challenging because of how rapidly these things are changing. All right. What does that actually correlate to in terms of what it's done to people in the United States? So I've juxtaposed a couple of graphs here. So the top one is daily cases that were reported to the CDC. The bottom one is daily deaths. And we're going all the way back to January of 2020. This is, as many of you may remember, this was actually us, right? We were first in the United States to report cases. So daily cases we see here, this was all original wild type. Saw a lot of deaths because we did not know what to do. We did not know how to treat people. There was no immunity, background immunity at all. So into then the end of the year, this is when alpha became predominant. Again, a lot of deaths associated with alpha. Increased infection potential. In other words, it was spreading more rapidly. At the time, we didn't think that it was necessarily more likely to escape. If you had antibodies, there was not increased immune escape from it. But then period of relative quiet, and then we came into Delta. And so Delta, the daily cases here, you can see here, not as many as when we had alpha, but much more deadly in terms of, for the number of cases per case, it was a very deadly virus to us. And then into the beginning of this year, this is Omicron. So Omicron, you may hear people say, and you may even think this, that it's not as deadly, which is true. It's not as severely dangerous for many people, but just because of the sheer number of people that are infected, you see here this huge spike in the number of daily deaths. Okay, then we got through sort of the first version of Omicron, and then we're into our latest version, BA5, and this was our summer here, and still relatively low, but again, lots of rapid changes to this. Okay. I've also juxtaposed here are the major changes to the vaccine strategies to date, right? So end of December, I still remember how emotional it was that people were able to get their vaccinations, right? This was, thank God we can finally do something. And we were in the middle of the terrible wave of infections and deaths at the time. So that's the first vaccine. September of last year, this is when our first boosters got approved. And again, correlation does not mean causation, but shortly afterwards, we started to see a drop in some of the stuff. And then Omicron happened. And in around April, we approved second boosters for a select group of patients. Okay. And that's where we find ourselves at the end of August, beginning of September. All right. What does it all mean to have these different subvariants, and what does all this, like Greek letters and all, what does all this stuff sort of mean? And again, I'm not an evolutionary virologist, but I'm going to try to simplify some descriptions of this. So each of these variants of concern, early on, there was a decision under WHO to name them different Greek letters, right? If you remember, we were talking about the South African version, we're talking about the Brazilian version, and for good reasons that became problematic, not the least of which was because it was incredibly stigmatizing to the fine scientists at places like South Africa that were doing us a favor in discovering these, and they would get stigmatized because they'd be labeled with a virus name. And so Omicron, this variant, has 5 sublineages, and we're going to refer to 2 of them frequently, with the BA1 and the BA45. Some things to know and understand about this is that each of these labeled variants, Omicron, Delta, Alpha, represents a significant branch point from the underlying earlier version of the virus. Okay, so once we see evolutionarily like a big split in it, then we have given them Greek letters as names. So it's not entirely the same, but if you think a little bit about like what you remember about flu and flu pandemics and epidemics, that you can think about like the shifting of the virus through within a given variant as a little bit of the shift that happens, versus these big changes in terms of the branch points as sort of like the big shifts in, or the big not the drift, but the shift in terms of what it means. So BA1 through 5, we've gone through those numbers. They represent small changes, but the numbers don't necessarily represent, like, it's not like BA2 led directly to BA3 or BA3 led to BA4. It doesn't entirely work like that. So the numbers can be a little deceptive in thinking about what that all means. The other big thing to know about Omicron, and as you all know, is that it was the first one really to get us very concerned in a large number, that not only were we seeing increased infection spread very quickly, but we were also worried about like that it was escaping some of our prior vaccinations and some monoclonal antibody treatments. We saw some of our monoclonal antibody treatments no longer effective at all against Omicron. We stopped using them because the variant was so different. Okay. This is complicated, and I admit that this is science that I don't understand entirely, but I want to very generally explain what is being shown here. This black dot here, so this is a graphical representation that's meant to try to depict for you how different evolutionarily and the conformation of their spike proteins are. It's meant to depict for you how different the virus's strains are from one another. So here was the original wild type version of the virus from Wuhan province. And then our first major branch point came with so we're talking about Omicron. So BA1, Omicron, this was a big shift, big change. BA4 and 5, this is the latest version of the virus, is very different in the way this is depicted here from even BA1. So there's actually a fairly big difference there, and it's a big difference from the original version. Very complicated again. This dark spot here represents the original version, and you kind of have to imagine like orbits here, like different orbits, and a 3-dimensional picture, that the various Omicron variants are related to one another, but that again, BA4 slash BA5 is very different from the rest. Now, I'm going to interchangeably use BA4 and 5, and the reason I am is because it turns out that the spike protein for BA4 and BA5 is the same spike protein. They're not different from one another. And so if I slip between those usages, that's because they're the same exact spike protein. Okay. Let's do a quick overview on what's been going on during the Omicron era. And so this was, I think it was Ruth who was talking about this. This happened in around May, where what we saw here, so the dark line here represents number of cases that we know about here. And the yellowish line here, the orange line here, represents among people who got tested, what's the percent positive rate for those tests? So these are the PCRs, these are the nucleic amplification tests. So we see here in the past, this is correlated very tightly with what we see going on in the community. This is the first time that we see this big separation between test positivity rate and actually the number of infections reported. And like many of you know, you may have even done this yourself, not everyone who tests positive on an antigen test reports it. There is a way, if you're curious, there's a way within the state to report your positive antigen test. I can tell you honestly, I don't know anyone who does this. And that's probably explaining here now we are a little bit blind as to what's going on in the pandemic in terms of the actual number of infections. We're making a lot of inferences from here on out because we have widely available home tests in a way that we didn't before. Okay, other trends. Since March, April, we had seen a lot of benefits for the vaccinations and a lot of benefits, particularly with the older group. But now we see a big split happening in the hospitalization rates of older people versus younger people. This may reflect your own experiences you see in the hospitals, like who's still coming to the ICU. Like in the early days, it's the older people who are coming in, those with a lot of comorbid issues. And so again, the comments that you hear a lot of people talk about how it's a mild form of the disease, true for many people, but still our older patients remain at most vulnerable risk for hospitalization and death from the Omicron variant. And that also correlates with what we see in the death certificate. So again, increases in death rates in the older adults relative to other groups. Not all bad news. The CDC has really taken a strong, hard look at equity issues, and the big differences in hospitalization rates and death rates and those sort of things were where there were lots of disparities in terms of those outcomes. Those disparities have narrowed by quite a bit during the Omicron surge. Now, is that because we're doing better, or is it because Omicron is so infectious it's affecting everyone equally? Don't know exactly, but it is perhaps one small bit of silver lining that some of the disparities and differences are less prominent during these last few months. Okay. We're gonna give an overview of about how vaccines are approved. And then at 10, we'll aim to take a break at 10 or a little before 10 so that people can stretch their legs. But then when we come back, as you listen to this discussion about how vaccines are approved, when we come back, you all are going to be placed in the role of the ACIP, the advisory group to the CDC. And I'm going to present you the same data that they saw. And we're going to put a poll up and ask you to vote on how you would decide about whether to approve bivalent vaccines. So as you think about this, as you hear about how vaccines are approved, I want you to keep that in mind and imagine yourself in that role. Okay. Complicated slide, but let me just walk you very briefly through this, which is how are vaccines approved in the United States? How are vaccines, new drugs approved, but especially vaccines? So normal flow of things is that there's a lot of basic research, discovery, and preclinical studies that can usually take years of work. And the analogy here is that actually some of those developments that I mentioned to you in making these vaccines more effective. Those had been happening over the last 5 or 10 years, even before the pandemic. Some of those discoveries were new, but we built on those. So the basic research, very important to all this work. Phase 1, phase 2, and phase 3 studies happen. And then there is a submission to the FDA for a biologics license application. So the FDA then takes the data, they approve, they can choose to approve the vaccine, but what they want before they do that is they want to review that with their advisory group. So the FDA has an advisory group. This advisory group is called the Vaccines and Related Biologic Products Advisory Group. This VRBPAC group reviews the information and says yes or no to approval of the vaccine. Once the vaccine is approved, it's basically the FDA saying, yes, the company can sell the drug, can produce it, can make it available. The CDC pulls together an advisory group, the Advisory Council on Immunization Practices, and that advisory group advises and says to the government, this is how we think that this approved drug should be used. So there's two parts to the safety part structure here. So the FDA is the one that's really looking at the manufacturers. The ACIP and CDC is looking at the consumers, the patients and individuals. And the two are working together, and they both have independent, separate review processes. Once FDA advisory group meets and makes a recommendation, the FDA has to sign off on it. Similarly, once the ACIP meets, the CDC director has to sign off on that. Then all that stuff that happens afterwards with monitoring is sort of jointly monitored by the CDC and FDA in terms of the safety. Okay, so that's in a nutshell the approval process. This is busy, I'm not going to show that. What is emergency use authorization? We talked a lot in the first 2 years of the pandemic about EUA, emergency use authorization. And so EUA was in response to 9/11. We just had our, 21st anniversary, or yeah, 21st anniversary of 9/11. So after 9/11, there was a law that was passed by Congress that was made because you may be too young to remember this vividly, but there was an anthrax mail attack scare. There was anthrax being mailed through the mail. And they wanted to produce a way for FDA to rapidly produce things that didn't take years to produce. And so they said that we're going to let you have an emergency use authorization. And so with the language of it says that if things are a threat and there's an emergency, like a pandemic, there's no other alternatives, and you have reasonable belief that it might be effective, you can shorten the process that it normally takes you. There still has to be proof of safety, and it usually has to have substantial evidence, but we're not going to hold you to quite the same degree of evidence than we might for full authorization. And so we used it when we first approved oseltamivir for H1N1 flu. We used it for some early biologics against MERS and Ebola and Zika virus. So it has been used previously in the past. So this is a graphic representation of, again, what I said, clinical trials. Oftentimes it can take months or years, and then it goes to this Vaccines and Related Biologic Products Advisory Committee to the FDA. Mac. Then FDA approves, and then it goes to the ACIP, the CDC approves it, and then it goes to the public. That's the normal regulatory flow of this. Emergency use authorization. Again, all this process is much faster and much more rapid. You've seen plenty of examples where the FDA meets and then literally the next day the ACIP meets. Normally that's a period of oftentimes 6 months or longer between those. And then the whole intent here is that once you're through emergency use authorization, after you've collected more data, then it goes back to the FDA and then gets full approval, which is in fact what's happened with the mRNA vaccines. Okay. All right. What has happened? What's going on with the bivalent booster? We'll talk about what the bivalent booster exactly means in a bit, but what is the process that has been in play? So in June, the FDA met. The FDA met its vaccine-related biologic products advisory committee with a question, which was, everyone knew at that point in time, the writing was very clear that we were going to need polyvalent vaccines. So not just the original version, but it was very clear that the virus was changing, the sand was shifting under our feet, that we needed to keep up. They met to say, what are the things that we need to consider when choosing which things are the extra versions of the virus. And they did something that surprised the manufacturers, and they did something that surprised a lot of people, which is they looked at the data, they looked at what was coming down the pike, and they said, we want you to produce a bivalent vaccine against the BA4/5 virus, again, same spike protein, not BA1 or not beta. Up until this point, Pfizer and Mrna were doing studies on a bivalent vaccine against BA1. That was the first Omicron variants, and so they said, okay, we're going to produce a bivalent version of this. Supply chain. It turned out to be hugely consequential because the European regulators, the Canadian regulators, all said to the manufacturers, no, we want the BA1 version. I'll show you the data, but they said this because we have data on BA1. And so the manufacturers were faced with a quandary, which was that we have to now split production because we have one very large and influential market demanding a product against BA4/5, and we have the rest of the world that's demanding against BA1. So it's going to turn out to be important. Okay, so they met in June. At the end of August, so just over 2 months from this, they provided to the FDA their data, and the FDA met. They did not do their advisory committee group, but the FDA itself met and said, yes, we are going to approve based on the information that you shared, and you'll see it soon. We're going to approve these bivalent vaccines. The next day, advisory committee met and said, I'm not giving away anything you don't know. They approved it. And by the 7th, people are getting vaccines into arms. Yes, that's me. And so if you think about this, and if you think about where we are, I just want to pause and appreciate for a moment just how fast this is, right? FDA saying to the manufacturers in June, that by September, we want you to have millions of doses of vaccine for something that we're going to make you change midstream from what you were planning, and to have people getting vaccinations into arms just over 2 months later. I mean, that is truly mind-boggling and remarkable to think about. Okay. Let's take a break. Because we've gone over a lot of information. There's still a lot more information to go through. But when we come back, you are going to be the ACIP panel, okay? And in the ACIP panel, you are going to be voting on this question, all right? Does the Advisory Committee on Immunizations Practices support the use of updated bivalent COVID-19 vaccine booster doses for those individuals currently recommended to receive a booster? Okay, so that's what you will be voting on when we come back. Okay, so let's take a 5-minute break and come back at 6 after or 7 after 10. Okay, we'll see you all soon. All right, let's get back to the question at hand, which is whether we should vote for these boosters. All right, I'm going to reshare this. Okay. Yes, hold a thumbs up. You can see my screen again. Yeah, good. Okay. All right. So again, here's the question at hand. How do you vote on whether to approve bivalent vaccines? Okay. And I think it's important to really actually have in mind what we are moving towards, what the CDC and what the FDA, what we're trying to move towards as we think about this. Right now, we are based on a system of dose counting as thinking about our boosters, right? Like you do this, and I do this in clinic. I go and open up and they see, okay, how many doses have you had? Are you eligible? Are you due for another dose? And that sort of thing. And up till now, we have gotten up to 4 doses for people aged 50 and older. we want to be moving, and what CDC and really everyone is moving towards is trying to get towards this idea of not dose counting, but really thinking about time since your last vaccination. And that we want to try to move this ultimately towards more of a flu-like model if we can get there, right? Like, so we were talking about like everyone having their primary series, whatever that looks like, and then thinking about like some sort of schedule about boosters. Okay, so that's the goal here. That's what we're hoping to move towards. All right. If you are the ACIP and I'm putting you in that role, what are the things that you would want to know? What are the kinds of questions that you would need to know in order to vote on this question of whether to approve bivalent vaccines? So unmute yourself, holler it out, throw it into the chat. What are the things that are crucial for you to vote on this? Anyone? Besides whether the microchips are fully powered and all that. Safety data, yes, absolutely, Roxanne. Like, is it safe? What are the side effects? What are the adverse events? Absolutely. What else do you want to know?
Like the underlying disease prevalence and things like that.
Yeah. So you want to know a little bit about what is sort of maybe said another way, what do you predict is going to be happening? What do you see is happening with the disease? Yes. How long immunity lasts? How applicable the results are to the general public. Luke, thank you very much for that. It turns out that the yes, that is important. We're going to come back to that as we talk about acceptance. Any other questions that you're wondering about that might help you answer this question? Those are all really, I think, useful questions and things to think about. I don't know if you can see it. Some of this got cut off on my screen, but I'm going to group some of this information into these series of essential questions that the ACIP had as they approached this. And so when I literally sat through 7 hours of their testimony in order to go through this in all their, like, I don't know, it was like probably about 600 slides. And what I'm going to share with you is sort of a distilled version of that. But these are sort of the essential questions that they had. Okay, so they started with what is known currently about the uptake of the current monovalent boosters, so the current version of the booster. So if we go there and said, what do we know about this? So here's some information about this, right? So primary series, we have fairly good uptake overall in the population of the primary series. But once you start breaking this out into different age groups, you see that this does not actually apply equally across all age groups. So in the most vulnerable, the 65 and older, we have very good uptake. We've approached really close to probably the theoretical limit of how many people are going to accept it. And we're, 85, 90% of those people. But as you go down in ages, you see here that, the younger that you go, the less likely you are to have completed this. And this one is alarming to many public health folks, that even though the approval came late, that we've seen a plateauing very early on, somewhere around a third of people who are eligible among the 5 to 11-year-olds. So school age kids, only about a third or so have gotten their primary series yet. Okay, so what about, but that wasn't the question. What about boosters? So for first boosters, again, the most vulnerable group in terms of risk and death, of severe disease and death, the over 65-year-old group, pretty good. But then if you think about the converse of this, that we have about a third of the most vulnerable age groups that haven't even gotten their first booster. And again, the numbers go way down as the ages go up. And for second booster, so this is only the older than 50-year-old group, we see here that close to 2/3 of people have not yet gotten their second booster at all. So real issues overall in terms of vaccine uptake and acceptability. Terrence, to your question about how does this compare with vaccination uptakes of influenza? So vaccine uptake in influenza, not necessarily that great either. I think that there's useful comparisons, and then I think some useful contrasts with the influenza version, which is I think that the numbers that accept it from year to year are fairly stable, right? Like you probably know this, it's often the same people who refuse it every year and the same people who accept it. This is a fairly dynamic thing that's going on here, and it's not exactly clear whether it's the same people who are refusing flu vaccines that are also refusing COVID boosters. A lot of this is still not quite known. That's my long-winded way of saying I don't know the answer to your question, and I'm not sure we know it yet. Equity. So how does this vaccine coverage rate and booster vaccine coverage rate compare across different groups? And you'll see that there are big differences, disparities in acceptance across different racial and ethnic groups. And I've circled one group in particular, the Latino population, where uptake is quite low in this group. And so I think this is not even the ages is one reason in which these numbers split out, but also by race and ethnic group, this also splits out as well. So here's sort of an overview summary of what we know about this. So lots of people, 223 million, 224 million people have gotten a vaccine with primary series. That represents 72% of the overall population. About half of people who are eligible have gotten a first booster dose, and only about a third of people who are eligible for a second booster dose have gotten a second booster dose. And there are, as we said, disparities, differences according to age, particularly among the children and by race and ethnicity. I didn't show you the data here, but also by disability status as well. Okay, so let's go back to those essential questions. Okay, so another essential question that the group asked was, okay, what do we know about whether the regular boosters, the monovalent boosters, the original version, are effective? Do we know that they're effective, and what do we know about that? And so for that, here are some of the data that they showed. And this is really, this matches some of the stuff that we already talked about, but this is a very busy slide, but let me share with you what is intended to be shown here on the slide. So this black line here represents essentially no difference between the placebo group and the intervention group, so zero effectiveness. And just look at the trend, ignore the individual lines because they've broken it out by age, but the overall trend is that time from your last dose matters a lot to the effectiveness against getting infected in the first place. So much so that by 3 to 4 months, I think most people, you should assume that most people have very little additional protection from the vaccine from getting the infection in the first place. All right. Now, again, maybe we're looking at the wrong thing in saying we're trying to prevent infection. And what about for more severe disease? So busy slide. On the left side here, this side here, this is what if you've gotten your primary series and a booster, how does that compare against people who haven't gotten boosted at all? They've broken it out by age group. But again, by 3 to 4 months, most of that additional protection has waned, and there's very little additional protection from even one booster. The good news seems to be that there seems to be a little bit slower waning of that protection that may happen with additional boosters. There are a lot of confounding factors here, not entirely clear that this will hold up, but there is at least a suggestion that more boosters actually may lead to somewhat slower decline in some of this, at least in some populations. Okay. This is what we care about, right? This is what does it mean in terms of deaths and incidents and all that sort of stuff. And so, you know, in June, so this is in the height of Omicron, people who are 50 and older, so those people who had gotten 2 booster doses or more, had a 14 times lower risk of dying than those who are unvaccinated. So unvaccinated group here, the dark blue line here represents people who've gotten primary series in 1 booster. And they were 3 times lower if you had 2 boosters, a second booster. So even with each incremental one, so the question was, is 1 booster enough? it is incrementally better. It's not as large a drop as we might see otherwise, but it is 3 times lower than if you gotten the booster dose there. Okay. What about people who've had infections, so-called natural immunity? How does that compare to people who've gotten vaccinated? How does that compare against people who have gotten prior infection and boosted? What is the benefits here? And so if you have prior infection here, I'm gonna, this is on the left here, this is Pfizer. On the right here, it's Moderna. I'll just focus on the right here. So if you have prior infection, your effectiveness against against symptomatic infection, about 50%. If you've had 2 doses, your effectiveness, again, this is after a number of months after your vaccination, has already dropped down to very little, but with each boost, you can get it back up again. The group that seems to actually have the highest protection, and we'll talk about what protection means, is actually those people who've had infection and got boosted. So in other words, hybrid protection, so prior infection alone is not equivalent to getting prior infection and boosted. That seems to be the best of all combinations in terms of your protection against future symptomatic infection. Okay, so this slide summarizes it. It says, effectiveness continues to be higher and more sustained. The efficacy during the time when BA4 or 5 was predominant was generally similar to earlier periods. 3rd doses seemed to add additional help. The coverage of the 4th dose was too low to draw conclusions. But so those are some of the information, some of the data around what we know about monovalent vaccine boosters. Okay. Now here's the important part. Do we know it works? What are the data that support the efficacy of the bivalent booster? All right. We're going to spend a little time talking about this. So some things to understand about the bivalent booster here and what is intended. So the bivalent boosters, so the original version of the booster is 50 micrograms for Moderna, 30 micrograms for Pfizer. They are starting with the same dose, so 50 and 30 for their boosters. But what they've done is they've split it between the ancestral, the original version. So half of it's the original and half of it's a new version. Why did they choose 50? Why don't they just shove all the variants in, right? Like, why don't they pack in beta, delta, all the Omicron viruses? The reason that they didn't do that is because they are choosing this dose based on what they see about the reactions. They know that increasing the dose increases side effects and reactions. And so they're choosing, here's the cap. And if we choose smaller and smaller slices, like to give you more coverage of different types, we also know that we're going to have less of a bump in antibodies as the size of the dose decreases. So that's why it's bivalent rather than trivalent or quadrivalent and those sort of things. You'll see here an important thing here, though, which is that the trials, the trials that they did were against bivalent boosters with BA1. And I said to you already that in June, FDA said to the manufacturers, no, we want you to produce this against BA5, BA4, 5. This caught everyone by surprise. And up till this point, Moderna and Pfizer were plugging along with their study and planning on showing data on bivalent boosters against BA1. This bears repeating because this is a huge issue here. There are no clinical outcomes trial data that was available for the ACIP nor for you for bivalent boosters that use BA4/5. Okay, no clinical trial data that's available. We do have mice models. We do have outcomes on the BA1 version, and you're being asked to extrapolate based on what they present to you on this other version of the bivalent virus vaccine. Okay. Super busy slide. So what I'm going to show you the Mrna data here. So Mrna was doing bivalent tests with beta. They were doing bivalent tests with BA Omicron, and they have recently started and have a study ongoing for BA4/5. So they don't have outcomes. It's still ongoing, and they know this. They shared this. They admitted this into as they talk to the ACIP. What they did though is they said, okay, what do we know about it? What have we seen? What are we looking at? So they presented, Pfizer and Moderna both presented their data on their bivalent vaccine that incorporates BA.1, the earlier version of Omicron. And so what they are showing here, what I'll show you over the next couple of slides, is they basically take serum from these individuals and they add BA.1 Omicron, and they look at to see sort of like how much is necessary in order to neutralize all this. And they're using this information to make inferences. This actually does correlate fairly well to protection against symptomatic disease. But so what you're looking at here is that if you look at all participants on the left, the light blue here is what if you give a second booster to just using the original monovalent vaccine like we've been doing. And then let's compare that to those who got the bivalent dose. And so what they saw is that, yeah, not unexpectedly, you have good protection against BA.1. But you also have, even with the original version, you have an increase in production, even if you use the ancestral one, to protection against BA.1, even if you're using something that's not specifically targeted against this. Is this hybrid immunity assuming that you're infected with Omicron or any COVID strain? That I'm assuming you referenced the prior figure. I'm not sure that those researchers broke that out, because one of the things that we know is that a lot of things that we can do in research settings, we're not doing broadly. So we're not broadly testing in terms of people's which strain that they got. I'm not aware if that study involved a test to see which strain of Omicron or COVID that they got. Okay, so that's protection against BA.1. What about protection against BA.4/5? So again, this is what became the circulating dominant strain. So they said, again, this is a booster that has BA.1, a different Omicron strain. How much protection does that booster give you over the original version against this different strain of Omicron? So it was statistically significant difference compared to a booster with the original version, but it's not, you know, for lay people, for people even who are not lay people, but like clinicians, not entirely clear what this means in terms of actual additional benefits. Again, we don't have outcomes data yet. We don't have like the large studies that say, here's how many thousands of people may have avoided infection by getting the bivalent booster. So making inferences. I won't dwell on this, but the Pfizer data essentially showed the same sort of thing, that they were using inferences based on the BA.1 bivalent booster, not a BA.5 bivalent booster. Okay, they did use mouse models. They did neutralization assays. They stuck stuff into mice and they said, okay, how much additional antibodies do we see by giving these viruses? And they saw that, yeah, if you give BA.5 specific proteins, mRNA, that you see an increase in the vaccine protection. What's really interesting about this, and one of the theories and one of the ideas behind giving bivalent or polyvalent vaccines is that you can broaden the immunity perhaps to not just the things that you're targeting, but because the immune system is a little bit messy sometimes, that doing that actually introduces the opportunity for your immune system to see different looks and actually, in its slight messiness, produce vaccine or produce antibodies that might protect you against different variants to Omicron that are still yet to come. Now, things I'll point out to you, that and this study here, again, all using mice models, if you count them, we're talking about 8 mice in each arm of the study. So these are not large mice data that they're using as they're presenting this to the FDA. Sorry, FDA and CDC. Okay, again, statistically significant differences. What this means in actual practice, not at all certain that we have that information. Okay. All right, so summarizing what data we have. We don't have completed human outcomes data from bivalent BA.4/5 boosters. Pfizer and Moderna presented results from assays, and they have 1,400 people who've received, between the two companies, who've received bivalent BA1 boosters. They saw that the amount of circulating neutralizing antibodies in the past have correlated with risk for symptomatic infections, but these outcomes that we're really interested in as clinicians still being collected. Bivalent boosters using BA1, statistically significant increase in BA1 antibodies and BA4/5 antibodies, unclear how much that difference is clinically significant and how it correlates to actual outcomes. Okay, mouse models do show some incremental benefits. All right, so that's what you know, that's what the ACIP and FDA knew in terms of the efficacy of this. I don't remember who asked the question, but what is known about the adverse reaction, so side effects? So here's what we know about some of the side effects. So if you've ever done the thing, if you've done the, I don't know how many of you do the CDC survey where you get a text on your phone and it says, do you have any side effects? So this is based on VSafe. What they found, I'll walk through it very quickly, that as time went on, the further you got from your original doses, the less likely you were to have side effects. So dose 1 to dose 2, we saw an increase in side effects oftentimes. Makes sense, right? Your immune system's revved up. Dose 3 and dose 4, so with more boosters, those are oftentimes months later, immune system kind of ramps back down again. And so the side effects generally are much more acceptable, much milder in that case. This is the Moderna specific information. The big ones, like very serious systemic effects, were generally not seen in huge numbers and were less than in the original versions. If you're curious, just because, if you're curious, the bivalent booster from Mrna, again, just it is true for the primary series. If you look at the kind of degree of the line here for pain, the bivalent booster for Mrna was higher than the bivalent Pfizer version. Again, the dose from Mrna is 50 micrograms total. The Pfizer dose is 30. So some of that may be responsible for that difference in outcomes. Okay. I don't know how many of you have heard this or have come across this issue. There is, in the mRNA vaccines, there is an increased risk for myocarditis and pericarditis. That increased risk is not distributed evenly, and it is very heavily weighted among men or boys. It is much less so in females, and the timing again matters here. What we're seeing with booster doses is that, so this is where it peaks. So the 16 to 24 age or 12 to 24 age group for boys and men, that's where it kind of peaks. The second dose is where we see it highest, and with the first booster dose, it is still there as an increased signal, but much less so. What that correlates to in terms of second booster doses and or this bivalent dose, we don't know. Early indications are that we don't see a lot of this, and we don't actually see it in the young group, the little kids, for reasons that are really not entirely sure, probably have to do with hormones in terms of circulating hormones, etc. But we don't exactly know. The cardiac outcomes, many people have said this, but just so we can say it out loud, cardiac outcomes for COVID infection, much greater than the risk for myocarditis and pericarditis after vaccination. And the myocarditis that's happening is very mild, mostly speaking, compared to the viral versions. Most of the time in 80–85% of cases, symptoms are gone within a number of days. By 2 weeks, they're gone in something upwards of 95% or more. All right. Oops, sorry, I jumped the gun. So the last question to ask is, what do we know about what's coming down the road? What can we predict depending on whether we give the vaccine now or later, or we limit it to certain people, or we make it more widely available? What can we predict based on this? And so CDC did some modeling studies, and very, very busy, but what they modeled here is what happens if we limit it only to people older than the age of 50. Okay, so an age-restricted strategy versus offering it to everyone who already is eligible for it. So this first column here represents the assumption that we're not going to see a scary new variant that emerges in the middle of winter. This one here on the right says, what if we do see a scary variant? And what they saw was that, again, the curves are very confusing to look at, but overall, they said that if we limit it to only those who are older than 50, we would see a higher hospitalization rate overall and a higher death rate. If we made it widely available to those that are all eligible now, we would see a 20% reduction compared to what we would see otherwise. All right, what about, and this came up in the discussions, what if we waited on more data? What if we said, you know what, we're not comfortable with the amount of data that we have now. Let's actually wait for some of the outcomes data to come back. Let's look at this. What happens if we do this in November instead of September? What would we see? And what they modeled, what they predicted was that we would see 140,000 more hospitalizations and nearly 10,000 more deaths if we were to wait versus if we were to do this now. Okay. Cost effectiveness, in the interest of time, I'll skip over this, but the strategy that they were advocating for would save a lot of money if we were to do this. Okay. A lot of assumptions, wide variability in their estimates, but there was reasons to think it'd be cost effective. Okay. I'm going to stop there for a second and and ask if there are additional questions or things that people are wondering about before they want to vote on this question. You now have at your fingertips essentially all of the data that the ACIP had in voting for this. Okay, all right. I'm going to skip some busy slides here. Okay. So one thing I need to say, CDC has advocated for co-administration of this vaccine with other vaccines. So they said, yes, it's okay to give with flu vaccine. It's okay to give with other things. The one exception that they've put in here, and it's only relevant now, is that if you have an individual who's received a monkeypox vaccine, that they are recommending waiting 4 weeks after that to give a booster. There's thought to be perhaps a slightly increased risk of myocarditis after monkeypox vaccination, and so that's why they're asking for that. A question in the discussion from Luke: is there any reason to think that there's something fundamentally different about this vaccine compared to the original mRNA vaccine, or is it really the only difference the mRNA code within the vaccine? Luke, essentially that's that. And that is what the manufacturers were saying. That's what the FDA was saying as they looked at this, that it's basically the same mechanism other than the code is different, which is how they got to millions of doses within 2 months. Can the bivalent vaccines be the initial vaccine for unvaccinated people? Great question, Ruth. That is not what they were voting on, and they were that question did come up. They don't have data on that, and so that is not approved. What will be going forward, well, let me just share this. Here again is the question. The bottom part is an important corollary to this. They are also saying that they're repealing, getting rid of the monovalent boosters entirely now that the bivalent boosters are available. So that would be what they're voting on, and the result of that is that they would take away the monovalent boosters. All right, let's launch a poll. You're the ACIP. Is the poll on? I can't see it from mine. There it is. All right. You're ACIP, vote. Yeah, I shouldn't have given the abstain option. There's some people who are sitting on the fence still. We'll maybe give it another 30 seconds. We're not tracking this, I'm not going to think any less of you if you're not sure.
All polls are anonymous as well.
Mhm. Yeah, if you wondered. All right, there's a handful of people who haven't voted, but let's go ahead and close it, and let's go ahead and share this. All right. 75% of people said yes, 5% said no, 20% abstain. We won't count those for the final version. If you're curious, this actually correlates fairly well with what ended up happening. So the conversation, the concerns raised by ACIP, the issues that were brought up, there was a lot of discussion. Almost all the members were concerned that no direct human clinical data on BA.4/5 results were in. The FDA countered, they were at the meeting, they countered that this is similar to what's done with influenza. Every year in influenza approval, that they actually, the scaffolding's the same, the details of the windows and the dormers and that sort of thing are different, but essentially this is what they do, they said. The BA.1 bivalent vaccine is going to Canada. So we have people just a few hundred miles to the north of us who will be getting the different vaccine, and to Europe. All right, so we have data on BA.1. They said, why don't we go back to the BA.1? There was concern about the language. The FDA and the approval says at least 2 months since your last booster. I've shown you, I hope, good information that says that actually waiting a little bit longer, because they're silent on how long. There's no language in this EUA, in this language here, about what happens if someone had an infection. The members of the group and the CDC has actually come out and said this, that they would recommend waiting 3 months or even longer, because that might help improve protection and that there are less risks of side effects for things like myocarditis after your either your last booster or last infection. There was a lot of regret that people were saying, you're taking off the monovalent, what about equity issues because they're not immediately available in clinics? What does that mean in terms of ramping this up quickly? And this is not the focus of our discussion, strong concerns about errors that could happen with how this is done. I didn't show you the pictures, but the bottles look almost identical with just some slight different colors and slightly different wording. And when they looked at the errors and vaccine reported adverse outcomes, administration errors was far and away larger than actual serious side effects. And so people were very concerned about the risks for errors. Okay, but what they found, what they did is pass 13-1. So not too dissimilar to the numbers that you all voted on. And I think that they shared a lot of this sort of stuff. I think we're out of time to talk about hesitancy, but I do want to share a couple of quick slides and leave you some thoughts about hesitancy and how you address this with your patients. There was a survey that they shared as part of the ACIP discussion. There was an online survey that said, that found that 72% of the respondents online survey said that, yeah, they would get an updated booster. This does not track with what we're actually seeing in the data around boosters. So there's a little bit of discordance with what people are saying in this online survey versus what actually we see with the prior boosters. The things that are more likely to be unsure, causes to be unsure or not saying that they want to get updated. So the belief that you have enough protection from your prior doses or that the updated booster is not effective, those are the strongest predictors for those. Again, as you think about talking to your patients, these are things that will matter as you think about talking to your patients. Things that were pro getting a booster. So the belief that it can help me spread to others, that the cases have become more severe. These were some of the stronger beliefs that correlate with more likely to get boosters. This was not surprisingly not evenly distributed in terms of different racial and ethnic groups. There were differences in terms of trust, and those were important as we think about acceptance and uptake. I'm only going to highlight this last point, that healthcare provider recommendation for those that were older, 77% of adults said that their recommendation by their provider was one of the most important reasons that they went and got the booster. We often think sometimes that we sometimes give up and say, you know, people believe what they're going to believe. But this tells me that the most important thing that you can do in these next few months in primary care is to really talk to your patients about this and to have honest and robust conversations about this, that you can move the bar as it comes to boosters. So I know I'm spilling into Ken's time, so I want to stop here and thank you all. If there are questions that I didn't answer, then please let me know and I'll try to answer them. This is the quick and dirty version of what is now available. Once you've gotten your vaccine, we're no longer saying 1 booster, 2 booster, whatever. Once you've gotten your vaccine, wait at least 2 months, probably longer, but then you can give the bivalent booster dose at that point in time. Okay? Questions? Last questions or issues? Again, Ken, if you're on, I'm sorry I spilled into your time. Maybe he's not on. Okay. Any other? Okay. Kayla, do you want to, since Ken's not on, do you want to give us a break or?